So today I'm going to talk about my previous research on Cryo-Milling Tethered chromatin conformation capture, which is called a CTCC in short. So before developing this technology, there are some questions to bear in mind. So because we have high-C and TCC, so why not bother to do a CTCC? So the question is, first, do cross-linking and SDS treatment generate artifact, and do formaldehyde cross-linking and restriction digestion probe the genome evenly? So to address these questions, so we have a proposal to use the Cryo-Milling technology, because this technology has been used by our members of our center to study the structure of the nucleolus, the nuclear pore complexes. So they discovered that the Cryo-Milling procedure can open up the nucleus without disrupting the structural integrity of the nuclear pore complexes. So we wonder whether this technique can be used for studying the structure of the genome. So the whole experimental procedure is, first we flash freeze the cells pellets, and then we do a Cryo-Milling, and then we do a cross-linking or a light cross-linking, and then we use SDS and the restriction enzyme to treat the powders, which is optional. Then we tether the particle to a magnetic bead, and then we do a end labeling and proximity ligation, and then we subject the material to a next generation sequencing. And the core of the experiment is to use a Cryo-Milling machine, which is a sealed, insulated sealed jar containing liquid nitrogen. Inside the jar, there's iron bars. Then we dumped the flash frozen cell pellets into the jar, and then fed the jar to a Cryo-Milling machine, and then the machine will rotate the jar. And in the meantime, the ball will smash the cell pellets into fine powders. And as you can see that from this gel, we can show that the genomic fragment generated by the Cryo-Milling process is, as you increase the time of Cryo-Milling, you can see that the genomic DNA is shared into smaller and smaller pieces. If you Cryo-Mill for 1.5 hours, you can see that the majority of the genomic DNA is shared to a size of around 200 base pairs. And using multi-angle light scattering, we can see that the majority of the Cryo-Milled particles has a size of around 100 nanometers to 1,000 nanometers, which is a proof that the cell is grounded into powders, fine powders. So after the sequencing, we can see that Cryo-Milling actually samples the genome evenly across different subcompartments, while nuclease, such as restricted enzymes and macrocalco nuclease, has preference at compartment A region of the genome over the compartment B region of the genome. While Cryo-Milling somehow can open up the nuclear and make the restriction enzyme, such as HanD3, samples the genome more evenly. So in accordance to this even sampling of the genome, we can see that CTCC shows increased signal in compartment B, but decreased signal in compartment A of the genome. You can see that we compare the contact map between high-C and CTCC at the same total reads level. We found that if you subtract high-C from CTCC, you got the subtraction. You see that actually CTCC samples more at the compartment B regions of the genome, while high-C seems to samples more at the compartment A regions of the genome. And also, we found that CTCC actually detects less trans features. In high C, we usually see a very rich features at the trans regions. Here I show the regions between chromosome 7 and chromosome 8. But with CTCC, you scarcely see any features there, and except for a few regions, it seems very less features there. So the reason behind this is just imagine that there's two kinds of pairs in the nucleus. Here we show A and B and C and D. Well, A and B in proximity, but not in physical contact, while C and D are in physical contact. In high C, both kinds of pairs are able to be detected. But with Cryo-Milling, because A and B are not in physical contact, they tend to be smashed into different particles, while C and D, because they are in physical contact, they tend to remain in contact after a Cryo-Milling. So I introduced a parameter. I call it the same compartment contact. So because in high C, from the contact map, it was known that there's at least 5 compartments in the genome, compartment A1, A2, B1, B2, and B3. And the genomic loci belonging to the same compartment tend to interact more within the same compartment. So here, take this bean for example, because this bean, the pairs that have reads 1 belong to this bean, and read 2 also belong to A1 are regarded as signals. This bean belongs to A1, and the pairs that have read 1 belong to this bean, and the read 2 also belong to A1 are regarded as signals. And the pairs that have read 1 belong to this bean, and pair 2 belong to other compartments, like A2, B1, B2, and B3, are regarded as noise. So if we aggregate these results and we plot it as a violin graph, we can actually see that we can see that A1, A2, and B1 are less resistant to Cryo-Milling process. And the graph shown in blue the results from dilution high C, while the graph shown in orange from CTCC. We can see that there is a significant drop of signal to total from the results from both from the comparison after Cryo-Milling, but B2 and B3 remain almost unchanged. And the same is true for trans interactions. For trans interactions, we can see that there is a significant drop of signal to total after Cryo-Milling, but the signal from B2 and B3 remain almost unchanged. So from this, we conclude that there's 2 kinds of interactions. There seems to be 2 kinds of interactions in the high C experiment. One is called we called it a hard contact, and then the other we call it a soft contact. The hard contact is resistant to Cryo-Milling, while the soft contact is not resistant to Cryo-Milling. And the hard contact probably represents a physical contact, while the soft contact probably represents a relationship of just closing space. So besides these Cryo-Milling stuff, let's go back to the previous question that I raised before we set out to do the experiment. So what is the effect of SDS on that is commonly used for the high C experiment? So we compare, because of Cryo-Milling, we can do the experiment without SDS. So we found that CTCC without SDS results in a very less interchromosomal contact and drastically increased fraction of data represents short-range contact and decreased long-range contact, which is very similar to a typical micro-C experiment. And in addition to the data distribution, we also compared the results of CTCC without SDS and CTCC with SDS with that of the micro-C experiment. We found that actually CTCC without SDS, it looks very similar to that of macro-C. You can see that the slope of the curve is very similar to a macro-C experiment, and you can also observe the oscillation of the curve, which is commonly seen in a macro-C experiment. But with SDS, you lose this oscillation feature, and you also see a shallower decay curve. And in close lookup, if you look up check the contact map, you can see that SDS actually facilitates long-range ligation in CTCC. So basically, I compared two results. One is with SDS and one without. You can actually see that the one with SDS shows more contact in a TAD. And there is another result I'd like to share here, and previously described by the previous presenter, is that we show that RNA is very important for contact longer than 1 kilobase. So if we do the experiment in this fashion, for one group we treated with RNAs, and for the other we didn't. So we found that the one treated with RNAs shows very similar ultra-short contact, but drastically decreased long-range contact longer than 1 kilobase, which means that our RNA is very important for maintaining the structural integrity of the nucleus. So to conclude, CTCC can detect genomic structural features shown by other 3C-based methods. And second, CTCC samples the genome more vividly and enriches physical contact, hence more suited to study protein-DNA complexes in chromatin interactions. Third, CTCC can be carried out without SDS. And fourth, RNA is important for maintaining the contacts with distance longer than 1 kilobase. And five, CTCC can be directly applied to tissue samples. And last, I would like to acknowledge the help from my colleagues and the member of our center, Frank Albert's group, John Anderson's group, and Mark Rao from the Laboratory of Cellular and Structural Biology from the Rockefeller University. I would also like to address my gratefulness to Nicola from Curious Institute for helping with the HiC pipeline, Peter and Nether for helping with the HiGlass browser, and Yang Jiang from Jiang Xu lab for helping me to learn Python. That's all. Thank you.